Good morning. My name is Aaliyah Court, and I'm very excited to be here today to talk to you about environmental DNA, what it is, and how we use it as a tool to monitor the recovery of the smalltooth sawfish. And to start it off, what's on everyone's mind is what is environmental DNA? And it's really the shiny new object in the scientific community right now. It's similar to what stable isotopes were 20 years ago. So this is a new frontier, and I hope you guys kind of learned something what we did with our project, and I'm excited to share with you. Next slide, please. Today I'm going to talk about the sawfish historical distribution versus the current distribution, how that defined our core range versus what we talk about a non-core range is. This will decide our study areas for the project. I'll go in deeper on what environmental DNA is and how we used it for our planning process and methods. I'll also share with you our preliminary results and the conclusions from that. Next slide, please. Okay, so the historical distribution can be found on the map below. It's everywhere highlighted in royal blue. This consists of the warm waters of the Atlantic and the Gulf of Mexico. Unfortunately, by the 1980s, the smalltooth sawfish experienced a population crash, and they lost about 90% of their home range. Next slide, please. So currently, they can be found boxed off and with that big red arrow pointing to Southwest Florida and the Bahamas. Next slide, please. I want to share with you, we'll take a closer look at Florida, and we'll define where their core range is in Florida and where we consider non-core. Next slide, please. So when we talk about our core range, it can be found here in the red box. It consists of Charlotte Harbor down to the Florida Keys. There are 2 designated juvenile critical habitats, one in Charlotte Harbor and the other in the Everglades. And this provides estuary and nurseries for the smalltooth sawfish. Now, when I talk about our non-core range, it's anywhere outside of this red box. It uses the sawfish hotline where the general public can call in and report that they have seen a sawfish in their local areas. And we found that this can be all over Florida. And so that raised the question to us that these could be potential areas for recovery. Next slide, please. So 2 places come to mind. If Charlotte Harbor were to have a healthy and thriving population, we would see an expansion range to the next major estuary system north of us, which would be the Tampa Bay. And also directly east of Charlotte Harbor, we look at Indian River Lagoon because there once was a documented historical thriving nursery there, and they share the same temporal seasons as we do. Next slide, please. In 2016, Save Our Seas Foundation came out with a request for proposal for a sawfish project. And that really excited our scientists because it gave us the opportunity to go outside of our core range to study potential areas for recovery in non-core waters. And so we had a grant funding for 2017 through a 2020 project. The 3 areas of interest that we chose were the Indian River Lagoon, which we sampled in July, Tampa Bay, where we sampled in August, and the Mississippi Sound, where our collaborators, Dr. Nicole Phillips and her grad student, Ryan Lehman, researched. But for the purpose of this talk, we're just gonna stick with our Florida waters. Next slide, please. All right, down to the meat and potatoes. So environmental DNA, what is it? It is any form of DNA, such as hair, skin, mucus, scales, that is shed into the media like a water column. And so this picture really explains it great because within that tiny little vial, you can see hundreds of different species with their DNA in there. Now, that relates more to a biodiversity project. And what we wanted to do, since we're dealing with the smalltooth sawfish, they are critically endangered. And so we wanted a species-specific project. And that's why we chose to use the most sensitive technology on the market, which is the Droplet Digital PCR machine. Next slide, please. For our project, to break it down for you, we identified our areas of interest. We had about a 2 to 3 day process of sterilization of equipment. We collected water samples. We filtered those water samples and ran them through the ddPCR. Now, this machine, it can break down your water sample in up to 20,000 droplets and run the reaction on each and every droplet to determine if there's a positive or negative DNA result. So this machine, out of any PCR machine, there could be 3 methods used for that. But like I said, we are using a species-specific project, so we wanted to use the most sensitive assay on the market. Next slide, please. Our planning process in 2017, we used a well-tested species-specific primer. We took the DNA from a smalltooth sawfish and we compared it to the closely related species to it, like guitarfish or other rays. And we picked out the most unique molecule for the sawfish so it would become a true positive. In 2018, we defined what a sawfish sample would look like. The picture on the right shows that we went to a known nursery in the Caloosahatchee River, took a tub of water, stuck a live sawfish in that tub and proceeded to take samples to see what it would look like on an output graph. Throughout that year, we also sterilized our equipment, collected our water, and filtered those samples. In 2019, we analyzed the previous year samples with the ddPCR. We collected additional water and filtered those samples. In 2020, we're continuing to analyze our samples in the ddPCR. We're writing our final report and we expect to have a published paper by the end of this year. Next slide, please. For our field methods, we use the Sawfish Encounter database to give us a direction on where we should sample since we had verified reports from those locations. So an example for the map on the right of Tampa Bay, we divided it into 3 zones, green, red, and blue. We had 10 sites per zone, 3 liters per sample, and for a total of 120 total samples. We also had 2 controls per field day, 1 lab negative to validate our sterilization process, and 1 field negative to make sure that we had no contamination. Next slide, please. For our water samples, we use a customized weighted water sampler with aluminum and steel that would go down to the bottom of the water column to collect water. Literature suggests that because of gravity and temperature, that DNA holds better at that depth. So that's why we chose to use this specific airtight container for that. And also sawfish can be found on the bottom. So once we pulled the water up to the surface, we were able to bottle it. Next slide, please. For our laboratory methods, we had a heavy 2 to 3 day sterilization process for surfaces and equipment. The picture on the right is just a few of many bleach bottles we used for a 10% bleach solution. We paired it with a UV process and an autoclaving. We also made sure to filter our water samples within 24 hours of sampling so that the DNA wouldn't degrade. We know that under Florida conditions, sawfish DNA can last up to 72 hours. So we wanted to make sure we were really within that 24-hour sampling period. We also selected a 0.8 micron pore size to ensure that no DNA could slip through. And then we placed the filters in ethanol vials to ensure preservation. Next slide, please. Our filter rigs look something like the picture on the left. Once the water was drained, we would roll up our filters in what we called a taco. And this may look like a very simple picture, but don't let it fool you. It's like watching paint dry. So there were some nights where we were sampling from or until 1 a.m. and then well into 5 a.m. Next slide, please. So when we use our Droplet Digital PCR, or when we ran our samples through the ddPCR, the ddPCR would break it down in up to 20,000 droplets and run the reaction on each and every droplet to determine if it was positive or negative. Next slide, please. It would then place it on an output graph. The positives were on top where it had amplified DNA, and the negatives were on bottom in gray. Next slide, please. So going back to 2018, when we had defined a positive from a known nursery, this is where we took water from the Caloosahatchee River and we took a sample, and the ambient water, you can see there's actually quite a few positives there. Next, we stuck a sawfish in the tub and collected at time 0, and it's like fireworks. You can already see the DNA being pushed up to the top, being amplified. And then at times 30 minutes, I want to emphasize that nearly every positive in that sample, or nearly every droplet in that sample is a positive. And I want to draw your attention to the bottom of that map, and there's nearly 0 negatives. Next slide, please. So our Tampa Bay results, in 2018, we had 30 samples with 3 positives. And in 2019, we had 30 samples with 3 positives. This was really influential information because we can send this out to researchers in that area and they could direct their sampling due to this. Next slide, please. In the Indian River Lagoon in 2018, we had 30 samples taken with 4 positives, and in 2019, 30 samples taken with 1 positive. Now, we are unsure of the size of the sawfish, but because we're in a habitat where you can find small juveniles, it gives the scientists high hopes that this could become another thriving nursery. Next slide, please. Okay, so when we go back to our output graph of a known nursery from the Caloosahatchee, we have many positives. There were 119 droplets out of almost 17,000. Next slide, please. So when we're dealing with an area in the non-core waters where we don't know if there could be a population there, and we're dealing with a critically endangered species, we don't want to miss that 1 positive droplet that we got in the Indian River Lagoon. So using the ddPCR really helped with this process because it was the most sensitive platform to use. Next slide, please. The DNA in the non-core waters for an endangered species, it's like finding a needle in the haystack. So this was a win for scientists, and it gave us a sense that this was a true positive because we know that under Florida conditions, a sawfish was in that area within that 72 hours. Next slide, please. Our conclusions is that environmental DNA, it works. The methods have been developed and validated through our sterilization process, our species-specific primer, and using the ddPCR. We use this as a tool to support field research and target our sampling. The Smalltooth Sawfish recovery team can use the eDNA in non-core areas, and these techniques are customizable for other regions. So if you're thinking about doing a species-specific project, please come see one of us and we would be glad to help you with it. Next slide, please. I'd like to acknowledge our funding sources, our field assistants for taking us out and navigating in the field, and our lab assistants for giving us a clean place to sample. Next slide, please. Thank you, and if you have any questions, I know we have 1 minute, so I apologize.
Oh no, that was just 1 minute until the Q&A, so you're doing fabulous. Thanks, Talia.
Okay, thank you.
Yeah, we're going into the question and answer period. We're going to have about 5 minutes here for Aaliyah to answer any questions that have come through Minty. For those of you online, if you wouldn't mind now entering those, minty.com. And the information is in the chat box if you need the code. It's also at the top of the screen now.
Okay, so the first question I see is, how is the eDNA database built and who contributes? That's a question for the geneticist that we collaborated with, Dr. Nicole Phillips. She worked hard for the species-specific primer for that and using the ddPCR. Next question is, will this method work for mammals? Yes. So anything that has DNA, there are land method, like terrestrial methods that you can use with soil and sediment. And then because we're using the smalltooth sawfish in the marine environment, we use a water-based. When you analyze samples, do you target sawfish or look at all species present in that sample? So because our project is species specific, we set up the primer to pick out that unique set of DNA sequence. And we tested it against its closely related species, so we wouldn't have any problems with that. And that's why it gave us a true positive. But there are other projects under biodiversity eDNAs that could do multiple species. Okay, so next question. So like Olaf said in the movie Frozen 2, water has memory. You said the samples need to be analyzed within 72 hours. How long does the DNA stay detectable in ambient water? So literature suggests that in the Florida conditions, because of heat and other factors, that the longest time period would be up to 72 hours. So you always want to do it as quickly as possible, just so you don't have any chance of losing that DNA or degrading. The next question I see is, how pricey is a ddPCR? From my understanding, it's definitely very pricey. Next question is, I'll come to this side. Since you are sampling from the water column, how do you adjust for river or tidal flow? That is where you find the DNA within 72 hours. So really, when you talk about the flow, the river flow or tidal system, that's going to be an physical oceanography question, and that could be done with more research. One of the next questions, if it can get scrolled down, please. Will DNA sample be influenced by disturbing the benthic substrate? No, it won't disturb the benthic substrate, because when we pull up to a sample site, we measure the depth and then we drop the sampler accordingly. So it's not touching the bottom, but it's just hovering right over it. And then, do you use volunteers in your research? During normal sampling year for the Florida Fish and Wildlife, we do take on volunteers, and we'd love if anyone would like to contact us, come and email us. We can get you set up during normal times. And then for areas where sawfish are expected to expand into like Tampa Bay, are regulations in place and managers ready to protect them there? So we do have collaborators from the Smalltooth Sawfish Recovery Implementation Team there. And so there are no regulations currently in place, but everything's under permit anyways. And then, are you sharing your data via open science? I'm not too sure what that means. Like I said, we are publishing a paper for this eDNA project. And if you want some more information, please come see one of the scientists, and email us, and we'd be happy to talk to you.
Great, thank you, Aaliyah. We are now at the conclusion of our Q&A. For any of you who have further questions, feel free to contact her with her contact information in the proceedings document. And we will go on now to our next presenter, Joellen Wilson with Bonefish and Tarpon Trust.
Thanks, Jennifer. Okay, as Jennifer said, my name is Joellen Wilson. I'm the Juvenile Tarpon Habitat Program Manager for Bonefish and Tarpon Trust. And today I'll be talking about habitat restoration with a purpose. And although we're looking at fisheries, as you'll see, it's mostly about the restoration